Ladies, gentlemen, colleagues, welcome to this session. This session is about being able to to watch a discussion, really a creative discussion between two of our beneficiaries here at the European Innovation Council and two external experts. In the field of health and biotechnology, I'm the program manager of health and biotechnology, Danos Arsimanoglu, and it is my pleasure to have you today in this session. It is, first of all, what is the purpose of this session? The purpose is to be able to see this interaction, and it's not only for us to say at the EIC that we have good projects, good companies, very competitive startups or really promising projects, but it's also for other experts to challenge that and to validate that and to give us even more credibility than what we already have. I'd like to say, I would like to share with you the following as an initial thought. European Innovation Council is known for its big size, is known for its sizable investments. And of course, it attracts a lot of attention for that. European Innovation Council is also known because it can support and fund from the very early stages, we call them Pathfinder, to companies that are already established and they are scaling up fast. So it's a long range of what you can fund. And also European Innovation Council is attempting and will be attempting portfolio schemes managed by program managers and new methods really to stimulate and support innovation. What is not known about Innovation Council, I believe will be, allow me to have it as a secret to reveal it at the end of this session and hopefully to convince you about that. So we have gathered a group of very successful people here, and allow me to introduce them. These people are, we have selected one project from, and one researcher in charge from our Pathfinder stage of development, the early stages, is Professor, I'm sorry, I forget the name right now, Francesca Scalconi, I think, right? I'm sorry for that. And we have together with us the co-founder and CEO of the Ilia Pharma, Evelina Vakeho. We have two external experts, the director, the CEO of the National Irish Biotechnology Bioprocessing Center in Dublin, Darren Morrissey. And also we have the Chief Scientific Officer of a VC fund, a Paris-based VC fund, which focuses also on Pathfinder stage of investments. So, and with that, and hoping to reveal the secret that I promised to you at the end of the stage, I'd like to pass the floor to Serge, who is the Chief Scientific Officer. Serge, over to you to describe, what you do and exactly the main of your organization, the main focus.
This nice segue into my presentation. So indeed, my name is Serge Pempfer, and I'm the Chief Scientific Officer of Eurocare. So Eurocare is a new European investment company. We are not a fund, we are an investment and operational company that has raised 50 million euros earlier this year in order to contribute to the launching and the financing of very early stage startups. And when I say very early stages, it's going as far as being part of the ideation process of those startups. We want to support companies that are highly disruptive in two spaces that we have elected as being our priorities. It's synthetic biology and it is a microbiome, and also everything that comes in between those two fields, because we do realize that there is a lot of overlap between synthetic biology and microbes and microorganisms. And we want to do that within an ecosystem that we are currently building with the partners throughout Europe. So we have already finalized 7 investments so far in 6 months. So I think it's fairly fast. I'm giving here examples of 2 of them in the microbiome space. Stellate, which is exploring the gut-to-brain axis in terms of microbiome, and we have also Novobiome, which is more of a platform company in the gut-to-liver axis. And then we have two other companies that I'm giving as an example here in the synthetic biology space. The first one is CoAve, which is working for gene therapy. The company is involved in inventing new adenovirus-associated virus capsids. And we also have another company called Omne, which is specialized in what we call XNA or xenobiotic nucleic acids. That being said, I would like to spend a couple of seconds on explaining to you how unique we want to be as an investor. You can see here and on the left side of the slide, the old-fashioned way of launching a company. We would be, in essence, we would be waiting for a scientist to come up with an invention or discovery. The TTO would secure the IP in terms of moving into a patent application, for instance, and then people would start to wonder how to transform a solution into solving a problem, and that's why a company will be created. A little while ago, it was realized that that way of doing was not really always successful, so people decided to proceed in another way, which was to start maybe with the identification of a market need or unmet need, and that was like starting with a problem and then building a team, building a company to find a solution. And that's where the concept of strategic innovation intelligence, which is dear to the EIC, come into play. And we at EuroCare, we want to push that a little bit further. We want to push the outside of the envelope a bit further by adding a layer or a flavor of artificial intelligence into the process. So we are building an AI platform with 3 modules. The first one is to help us detect trends and tendencies in the market and help facilitate the ideation process. And then we have a second module that is going to help us identify in Europe those scientists that are the better equipped to help us co-build together a new innovative solution to the problem mentioned. And then we have the third module, which is more like a marketplace for the ecosystem. And that's what I call strategic innovation, artificial intelligence, so to speak. So very briefly, if we were to continue our interaction with the project leaders that we have in front of us today, I would say that as a EuroCare, we would certainly be very pleased to, first to invite them to compete for the next pitch contest next year that we organize on an annual basis. We certainly would be happy to consider them for our studio program. We would provide them with advisory support in technology and business development, and maybe consider them for an investment, either pre-seed or seed equity type of financing. As far as myself, I'm concerned in helping the project leaders. I have just to remind the audience that I've been the CSO of a stem cell company that was targeting diabetes a while ago. I've been the project leader of a new startup that was involved in engineered bacteria. I've been the CEO of a startup incubator for 15 years. And also I have experience in corporate governance in biotech companies, which I would be happy to share with the companies emerging from the EIC programs. Thank you.
Thank you very much, Serge. You already realized that they are doing investments in Pathfinder at Pathfinder level. That's very relevant to us. And they're high involvement in cell and gene therapy and in engineering. I will pass the floor to Darren Morrissey, who is the CEO of the National Iris Bioprocessing Center. Darren, over to you. Our second external expert.
Thank you, Danish, and thank you to the organizers for the opportunity to participate today. You can all hear me okay? I'm going to look at heads nodding. Excellent.
It's very good.
So as you said, my name is Darren Morrissey. I'm the CEO of NIBRT, the National Institute for Bioprocessing Research and Training, which is in the picture behind me. I'd like to share with you a little bit about the work that we do and how we're relevant to the European innovation ecosystem. NIBRT was established around 10 years ago with Irish government funding through IDA Ireland, which is our state agency responsible for promoting foreign direct investment. And they invested over the last 10 years about 70 million in this facility. to 70 million euro. And we are one of 6 national research and training institutes that are important from a strategic perspective for economic development in Ireland. So that includes biopharma, microelectronics, high performance computing, and other areas like that. Now, the facility that you see behind, it's 6,500 square meters, focused on pilot scale biopharmaceutical manufacturing. And we deliver GMP simulated environment where we can do upstream work, downstream fill finish. We have analytical facilities and process utilities as well. And really in these facilities, what we deliver is commercial training for our client companies. We work with companies on collaborative research. And then we also do competitively funded research, which we win from state and EU agencies. So everything we do, it's focused on the needs of the biopharmaceutical industry. And over the last decade, we've been working with most of the large multinational biopharmas, as well as then increasingly smaller biotechs as well, and helping them actually to establish facilities, manufacturing facilities in Ireland. So we've been quite central to the success story in Ireland of attracting these big companies and creating tens of thousands of high value jobs. Now, over the last 10 years, we've mainly worked in large recombinant proteins and monoclonal antibodies. I mean, that has been the focus of the industry. And we've built on a core foundational strength of protein characterization, analytics, glycan analysis, and so on. But over the years, we've developed expertise in other areas as well. And we are certainly broadening increasingly into areas like gene therapies, non-viral oligonucleotide-based therapies, which are of course increasingly relevant with the growth of mRNA as an opportunity, and then also cell therapies, both autologous and allogeneic. And this is where certainly I overlap in our interests with the work of Francesca and Francesca's group and company in the cell therapy space. So moving on, I mean, the services that NIBRT specifically then provides are focused on optimizing biological manufacturing processes. And we focus on things like molecular characterization. We work on cell technology and engineering and research that improves the yield from your biopharma processes. We work on research that improves stability of the end product, improves purity and quality of the medicine ultimately. And then we also are increasingly moving into the area of pre-GMP scale-up services. So we have a complete set of bioreactors of scaling size and capacity, and we can generate biological materials, and we can optimize their workflows, and we create new standards, which are of course particularly important for your new modality therapies. I think it's fair to say the largest part of our business is in the training area in bioprocessing skills development. We train and we educate the majority of people in Ireland who move into the industry, from manufacturing operatives right through to PhDs. And of course, we're also now increasingly training people globally as well, because biopharma talent is a global challenge for everybody. And then we provide the biopharma community with access to cutting edge infrastructure. And I kind of highlighted this because this is actually really important. It's built on very close partnerships that we have with an array of biotech and bioprocessing equipment vendors. Some of the logos there on the right of the slide. It's not an exhaustive list. We pretty much work with everybody. And that's really important, I suppose, to create NIBRT as a test bed for the biopharmaceutical industry in Ireland and in Europe. So one last slide, just to conclude. I did really want to, I suppose, share with you my views on where I think the opportunity lies for Europe. And that is in the rapidly evolving area of advanced therapeutics in biopharmaceuticals. There was certainly a time when biopharma was really only about big players, big multinationals, European, US, Asian, producing large molecules in large quantities for large cohorts of patients. And frankly, this modality, albeit very profitable, was quite a homogeneous environment, very little diversity. One manufacturing facility looked very much like the other. And the innovation was quite incremental. So in 25 years of that industry developing, there really were no major leaps, great leaps forward. Lots of very solid incremental innovation though. Now, the major growth projections are focused on this new wave of advanced therapies, cell therapies, gene therapies, viral and non-viral, oligonucleotides, and so on. And with that comes rapid innovation. And that's underpinned by convergence with technology as well. So digital-based technology, materials-based technology, data analytics, and so on. So I see that really that there's many multiples of players now in the mix. So you've got your large multinationals, but you also have your SMEs, your startups, you have traditional biopharma, but also cutting edge biotech companies and genetics-based companies. And then you have your technology players, and then you have your big data players. And in this environment, I think real high performing partnerships between these players, and that includes government, by the way. So it's private sector and public sector. It involves academia as well. These high performing partnerships are absolutely critical to help companies.
Thank you very much.
Yeah. Great. Okay. Well, I will just one last sentence.
Please.
I think the nub of the issue for me is I think that the European Innovation Council has the opportunity and the capability to broker and catalyze these high performing partnerships. And I think creating that environment for innovation intelligence. And I do think where we're located in Dublin, east coast of Ireland, it's kind of a microcosm of that type of activity.
Very informative. Thank you very much, Darren. And I will quickly pass the floor to Professor Spagnoli, Professor of Regenerative Medicine from King's College. I'd like only to say about Darren's presentation, there are very few places in Europe that they have the training capability and the research and the bioprocessing knowledge and skills that they have in Dublin. So this is already something that we are putting, the people and the skills together, and we're very excited about that. So over to you, Francesca, for your presentation. Francesca has been founded by EIC. Thank you very much for being with us today.
So first of all, thank you for the invitation to speak at this EIC summit, and so it's a real pleasure to be here. In these few minutes, I will just give you an overview of my research interests and then tell you a bit more about what we are doing in the context of a consortium, a FET-open consortium that I'm coordinating. So in my lab, we study how organs are built and how we can reuse this knowledge for regenerative medicine. So we start from a developmental process, we build knowledge and platform, then for investigating, for understanding how organ works, and for modeling diseases, and for generating therapeutics like cells or tissue for cell replacement therapies. So we primarily focus on the pancreas that you can see here in these pictures. So this organ controls vital functions in our body. So these islets that you see in green in the center of the picture are fundamental, are responsible of our blood glucose metabolism, while all the cells around in red are the acinar tissue, which control our food digestion. These two compartments are targets of still incurable diseases. Diabetes, as many of you know, is the result of a loss or dysfunction of the insulin-producing beta cells, which are sitting in these islets that I showed you in the previous slides, while pancreatic cancer mainly targets the acinar tissue, and this is one of the deadliest malignancy with basically no therapeutic options so far. So there is a clear unmet need here for better disease models to understand the biology of this cancer. With respect to diabetes, there are more therapeutic options. For example, islet transplantation is already a clinical practice, and it's considered to be a functional cure for diabetes. However, here the limitation is the scarcity, the very little amount, the limited amount of islet available from transplantation. So the need here is to establish a renewable source of pancreatic islets that could be used for transplantation. And one of the most promising sources are the pluripotent stem cell. So in my lab, we study how these distinct cell types are formed during normal embryonic development, so in real life from a stem cell. We have invested quite some effort on defining autologous renewable source of pancreatic cells. And a more recent challenge that we have undertaken is on making more and better pancreatic islet cells. Better means more functional. And to achieve to do so, we have to focus our attention on actually the cells which surround the islets. So the islets are never alone during their life, and these surrounding include, for example, the immune cells, the fibroblasts, but also the vessels. And so to achieve this goal, And we reasoned essentially that if we were able to fully mimic this surrounding environment, this would promote the more mature, the making of more functional, so better islet cells. So to do so, we also reasoned that we needed a more innovative technology and an interdisciplinary approach. So we joined forces. We created a consortium founded by within the FET Open scheme that we call it PAN3DP. And the overall aim of this consortium is to bioprint pancreatic tissue for biomedical application. And why we chose the bioprinting? Because we reasoned that this technology enabled to combine together these different cellular components, so stem cell-derived pancreatic cells together with the supporting cells, and very importantly, to integrate the vasculature, which is fundamental in biofabricated tissue. So these are and then the advantage also of the bioprinting is the reproducibility and the scalability, which will be fundamental for future clinical application. So these are the two main aims of the consortium. So we started essentially by developing the technology and the bioprinting approaches of stem cell-derived pancreatic progenitors together with the with the supporting cells and with the vasculature, which have not been established before. And these are the different type of technologies that we have tested, that we are using, and different biomaterials and the different conditions. And this is just an example of the outcome. So these are like the prototype of our bioprinted tissue, in which you can recognize the different pancreatic cell types organized in the proper architecture and surrounded by a network of vessels. So this is very exciting. Essentially, it opens up now to multiple opportunities. It paved the road for engineering human pancreatic tissue that could be used for modeling pancreatic diseases that I introduced before, but also for drug screening, and in the future also for cell replacement therapies of diabetes. So we aim at generating vascularized mini islet. And I stop here by thanking you for the attention, and these are the different partners involved in our PAN3DP consortium. So thank you.
Thank you very much, Francesca, Professor Spagnoli. That was very informative and pretty much on time. And I hope as we're moving along that you understand the relevance between the external experts, and that's on purpose I didn't say much from the beginning, with our beneficiaries. And now we're moving from the discovery phase, where Professor Spagnoli has done a lot of successes and has been funded by EIC, to Ilia Pharma, and you will hear from the CEO and co-founder of Ilia Pharma, Evelina Vaikeho. Evelina, the floor is yours.
Perfect. Thank you so much. And very happy to be here enjoying this summit. So in Ilia Pharma, we develop immunotherapies using microorganisms to deliver stable therapeutic proteins. And in essence, we've developed a technology platform enabling the use of immunomodulators that are not suited for development as biologics, being too unstable, or as gene therapy, where the spatial distribution and dose is very important. And we have solved this for immunomodulators, chemokines, cytokines, interleukins, by generating strains of lactic acid bacteria, able to produce human variants of these therapeutic proteins that you see to the left. And today we've validated 6 different drug candidates and also built reporter strains. And at the moment, we have developed 2 drug products, a topical and an oral, that you see here in the middle. And going forward, we can address different diseases of skin and mucosa. And there to the right, you see examples of this. So here's an illustration of different modalities for microbiome-based diagnostics and therapeutics. And these genetically modified bacteria expressing human therapeutic proteins are classified as ATMPs in Europe and under gene therapy regulations by the FDA. So similar to CAR T cells being in vitro gene therapy. And today there are 4 different companies in clinical development of genetically modified bacteria expressing human therapeutic proteins. With our first project, we've proven accelerated time to wound healing in the skin, which is the primary endpoint that needs to be met in phase 3 clinical trials. And we've done this in mice, in mini pigs, and in humans. And in humans, we've also showed a 30% reduction in scar volume at 3 months. For the oral product, the scientific co-founders of Elia Pharma have just published a mechanism of action for where the bacterial strain that we use as a backbone in the ILP100 interacts with the immune system in healthy and colitic animals. And the key points from this paper is that the site of interaction is at the level of the Peyer's patches, the lymphoid tissue in the distal part of the small intestine. And this is producing downstream effects in the colon. So in Elia Pharma, we have proven that we can treat manifest colitis in experimental models. And there you see the ILP100 in green, as well as limit fibrosis development and increasing the number of anti-inflammatory and restorative immune cells in the colon. We've also shown that the effect size is about twice the size as the best variants of the 2 approved products, anti-TNF-alpha inhibitors and alpha-4 beta-7 integrin blockers. And we also recently completed a regulatory GLP repeated dose tox study. And this project is now fit for clinical development. So this is our current portfolio, where the topical product is developed in 2 indications and the oral product in 3 projects. We have an extensive support from the EIC for one of the projects. We do address large patient populations. We measure them in millions with growing markets. And we aim to make a significant impact for these patient populations, the healthcare system, and also make a difference in limiting spread of antibiotic resistance, and at the same time reward our shareholders. So this is the timeline showing our funding and milestone to date, and also what we plan forward. And we are closing a 9 million round with the EIC as an investor, the EIC fund of 9 million at the end of the year. So we have an 8 million secured, and the round is still open for 1 million. We have a seasoned management and board of directors, and the management have successfully accelerated development of biologics and ATMP products from preclinical to market approval. And the board have very complementary networks, and we have Patricia being one of our first coaches from the Horizon 2020 program that we're very happy for. We are backed by different EU initiatives, and we have definitely de-risked the developments, allowed us to keep a very high pace in the development, and provided a network to experts and facilitated private investment into the company. And I'm very happy to connect, and we're always looking to work with and learn from the best for our next stage of growth. Thank you very much.
Thank you, Evelina. This is a classical example of successful companies, successful in achieving the milestones, in getting the product, in getting finances from us and from investors. And also thanks to my colleague Robert Casals, he reminded me, Evelina was a finalist in the Women's Innovators Prize for 2020. So we are having very good people here presenting today, and we're very excited about that. We are getting into the second part of this session, the more interactive one, I would say. What we'll hear from now on is you will hear questions, challenging questions from the external experts to our beneficiaries. And as you saw, as you realized, even for people outside of health, we are trying, I hope you can see a little bit the relevance, at least for those outside of health, between the experts, their skills and expertise, and our beneficiaries. So first, I'll give the floor to Serge Panver, Dr. Panver, to ask a question to Francesca, I guess, right? Yeah. Serge, over to you.
Yeah, thank you. Well, Francesca, first, congratulations on your receiving this support from the EIC, which I guess is extremely important in terms of leading innovative projects to the market. And about the market, that's exactly the point I would like to make as an investor, that for us, a product, be it a beta cell graft or anything else, well, will only be useful if it is adopted by the end users, in this case, let's say the diabetic patients. And the point I want to make more precisely is that I believe that diabetic patients don't care that much about how their graft has been designed or has been assembled or printed. What they have is a list of requirements, and they want any new product to meet those needs. And it's the fact that the graft has to be glucose responsive, that the amount of insulin secreted has to be substantial, that the device or the graft has to be functioning for a long period of time, and I think we are talking years here or even more, and also that the treatment is affordable. And I think another very important point is that diabetic patients don't want to trade their insulin injection for immunosuppression. I believe that nowadays nobody wants to be immunosuppressed in any way or for any reason. So yes, I believe that I would be really interested in hearing from you how all these elements, this patient wish list, so to speak, which is also a market requirement in a way, have been incorporated from, or I would even say anticipated from the start in your project and in your consortium.
So yeah, thank you, Serge, for the challenging question. And yeah, I agree. The patient wants a product that functions and that is safe. And I can say that to make such a functional product, this requires a full understanding of the biology behind. So in a way, the example of my field of the pancreatic beta cell replacement field is a very good example, because it's really the little success that we are seeing now in going into clinical trial is really the result of the past, relatively recent actually, the past 15 years of research, of very active research in the field, because the first promising product that went into first clinical trial, it was a few years ago, about 2014, and these are islet progenitors which are generated starting from human embryonic stem cells, exactly with the protocol that I showed in one of my slides. And then a more recent and even more successful example is just a bit less than 2 years ago into clinical trial, in which the scientists actually decided to wait a little bit longer to further figure it out, how to mature these progenitor cells into more differentiated and functional cells, as you mentioned, that you could, as soon as implanted, they are ready to work and to function and to respond to the glucose. And this is the results are very promising. So it's true. The other important aspect in diabetes, of course, and in this type of approach, is the immunosuppression. And of course, the final goal is to also provide to spare the individual transplanted with lifelong immunosuppression. Field have been active on that, and with, for example, encapsulation. So devices, there is a lot of studies ongoing on encapsulation, and there are also some limitations on that. And I will come now to how our approach actually could help that side, for example. And then, of course, the more kind of futuristic way is now the idea of gene targeting, and essentially being able to use directly stem cells, which is evading, so using the CRISPR-Cas technology, able to evade the immune system. So with respect to our technology and what is different, would be different in our product, I think the first thing is the vascularization. And it's has been clearly shown in animal model, at least, that the transplantation of islets with already some preformed vascular network, not only islet, but also stem cell-derived islet cells, it enhances their functionality and prolongs their survival. So in our product, the vascularization will be there from the beginning. And this type of then bioprinted structure with the vessels could also be essentially embedded, implanted within an encapsulated device, because the encapsulation, the point is that to essentially create a physical barrier, so you don't want that the vessels get in, because then you don't want that the immune cells also get in. But if we provide a product which is already vascularized, this would, enhance the survival, for example. And then, of course, we could also think about applying incorporate into our platform edited immune stem cells which are capable to escape the immune system. And then with going back to our product, of course, the other advantage is the reproducibility that the bioprinting essentially machine would enable, because this will be fundamental then for clinical application, and it's still a limitation in many of the stem cell-derived products, essentially.
Thank you, Francesca. It's very difficult for me not to engage in the discussion, but I have to try and have a different role here in such a field. Serge, please go ahead with Evelina is very ready to take up your question.
Well, first, let me thank Francesca for this wonderful explanation that she has given to us in reaction to my provocative question. Now it's indeed your turn, Evelina. Well, again, as an investor, I must say that we keep a keen eye on about 10 companies over the world that are in the engineering bacteria strategy, especially for therapeutic reasons, but not all of them. You have mentioned a couple of names, and there are also others like Synlogic, Chain in the UK, there is Novum and a couple of others. And again, as an investor, we are fortunate enough to receive the business plans of a fairly large number of projects or very emerging startups that want to jump into that field of engineered LBPs or engineered bacteria. And one thing that struck me really is that every one of these companies seems to have their specific bacterial strain, I would call it a workhorse, that they want to develop their product on. So it could be E. coli, it could be Clostridium, it could be Lactococcus, Lactobacillus, but every company seems to have their own reasons to have selected one strain or one species over another one. And for me, this is not an innocent choice, because the chassis, this workhouse, is really core to anything that has to do with either safety and efficacy. I mean, in terms of biocontainment, for instance, depending on which strain you are going to work with, I mean, it could be very different in terms of how the bacteria would behave in its environment, what would happen if it escapes that intended location, still inside the body and even outside of the body. So there are many questions that regulatory agencies are going to ask facing your choice for bacterial strains over others. And my question would be, first, I mean, how do you justify the choices that you've made? And also, if you have already asked and maybe received from a health agency a scientific advice or a green light to move forward with the strategy and the strains that you have decided to use as a chassis.
Thank you very much, Serge, and you are giving a very broad number of aspects for this field. And what I can say for Ilia Pharma is that my background is in immunology and physiology. So we had great knowledge of these chemokines, cytokines, and interleukins, and I have recorded their mechanism of action in vivo, and I'm very sort of satisfied with being able to describe new mechanisms of action in regenerative medicine. So we came from that angle, and we wanted to explore a cheap and scalable way to deliver chemokines. to treat disease of skin and mucosa. And we've done a platform with different strains of lactic acid bacteria, so not limited to one specific strain. So we've used only non-human strains of Lactococcus and different lactobacillus. So for the skin application, I would say the choice of strain is, the most important thing is that it's producing lactic acid and that it's a non-human strain that lives there for and delivers the whatever therapeutic protein you want for 1 to 2 hours. And that is what we consider a safe way to work with these bacteria. There is a good reason why we've started with a topical program and then that the oral program is our second. So we've de-risked the oral program significantly by starting a topical, because it's very intuitive and it's been easy enough to explain to regulators. And we've, during the time, built tremendous amount of safety data from skin samples, fecal samples, environmental safety samples, and so on. And we've had multiple regulatory interactions in EU and with the FDA, and we had it for many years already. And next up, we have aligned with the regulators for a phase 2-3 trial to be started next year. And we've done it by building extremely good data in-house and the rationales explaining how we see it and how we would like to have the clinical development and the CMC development aligned.
Thank you, Evelina. Thank you. I think you covered it all. So he won't dare again to ask such a broad question. He got all it covered. Just for the sake of time, Darren, please, let's try to be a little bit more brief in our questions, so to give more time to have more questions. Thank you very much. Go ahead, Darren, please. Francesca first, and then...
Yeah, to Francesca. Thank you, Francesca, for really what I think is highly disruptive leading edge research. And as you said, Danish, it's in the discovery phase. However, I suppose being the manufacturing person that I am, that'll be the nature of my question. So I'm conscious that pancreatic diseases, diabetes, pancreatic cancer, I mean, they're very widespread and diverse, dispersed patient cohorts. And in that sense, I'm imagining a large-scale manufacturing, widespread delivery kind of system that would be required. But then on the other hand, it is an autologous, highly personalized medicine as well. So I'm just wondering, have you given much thought yet from your discovery phase to the manufacturing strategy for these highly disruptive regenerative products? And the things for me that kind of come to mind are, is it centralized, is it large scale, or is it more decentralized, closer to the patient and closer to the clinic? Does it involve partnering with a CDMO or a vendor company or some alternative? And also, have you considered things like the complex logistics management chain of identity challenges that are around autologous medicines and therapies? So I think that the nub of the question is a manufacturing strategy. Have you considered it?
Yes. So yeah, thanks. And I think as you have seen in my presentation, we are really at the discovery stage, and essentially what we're doing is at the moment is reproducibility control and benchmarking and defining. So it's very early phase, but thanks for the question and for bringing this into the plate. And yes, we are aware about the importance of start thinking about the manufacturing and to think about a suitable platform essentially to really manage the multitude events that will be essentially how to maintain really this chain of identity throughout the supply. And this is critical either if we go for an autologous or for an allogenic cell therapy is really the fundamental aspects of the traceability of the product essentially. So we have to go back to more specifically to your question about decentralized or large scale. I think there is more and more like the idea, the attention on increasing the attention on there is more and more attention paid on actually decentralizing production facilities for gene and cell therapies product. So these in particular for a bioprinted approach would be maybe more suitable, I think, to our product. And this would be also maybe more, they would be also more beneficial for the traceability side. But I have to say that we are not going to be able to set up and decide this within our consortium. So this will be outsourced, and we most likely via a CMDO, essentially CDMO. So this is so we need for that essentially support for the early clinical development. So one advantage that we have in the context of our consortium is that we already have an industrial partner. So the company Poietis, they have a GMP grade already facility to bioprint skin for transplantation. So they are kind of familiar with this type of thinking and approaches. And we are willing to start discussion also with the cell and gene therapy catapult platform, which is actually, we have a strong advantage because it's physically located at the Guy's campus, so in my building. And so they have helped already, like this type of bridging scientific research with the full exploitation and commercialization. So for example, they have helped other for automating IPS manufacturing, for quality control of IPS differentiation. So this is the type of approaches essentially that we're thinking. And now I think it was a good occasion that I also meet you. So we might continue this type of discussion in the future, essentially. So
many
later
thanks.
on. So thank you.
That was very clear, and many thanks, Francesca. Clear message on time, perfect. Darren, over to you again.
Well, Evelina, another typically manufacturing-focused question, but for me, I suppose I'm coming from the standpoint of running a research and training institute that's focused on making large, complex proteins in engineered mammalian cells. So clearly, in bioreactors, it's a different gig. But I did have back in my career time spent as a postdoc working on gut microbiome, so I'm very interested in the area on the immunological aspects of the gut microbiome. Now, I'm looking at your product platform, and clearly it is a natural bacteria-based therapy, albeit engineered and curated. But all of that said, I mean, there's a lot of associated underlying heterogeneity between individual microbes. And I'm just wondering, what thought have you given to the manufacturing process in terms of kind of consistency and quality and purity of the product? And linked to that, and I think linked to Serge's question as well, the discussion with the regulators in that regard. I would just be fascinated to understand more how the regulators are engaging on that question of consistent manufacturing.
Yes, thank you so much for that. And I mean, we've hired extremely competent CMC people already from the start. We've manufactured more than 50 batches in-house. And we have a similar facility to Nybert, but not maybe as large, at the Cytiva site in Uppsala. That's called Testa Center. So there we've been going in before every tech transfer to CMO. So we've done the development part in-house, actually. But for each contract manufacturing, we've done the whatever GMP process in full scale just before the tech transfer to de-risk. And that's been extremely important. And as we've manufactured so many batches, we have data on scale effects that we don't see, meaning that we have good data on the consistency of the manufacturing process. And I would say that from a regulatory perspective, we're very well aligned on the bioprocess with the phase of development. And where I see or where we have invested a lot the last years, some things that never produces any milestone for shareholders, but that is into the bioanalytical development and GMP validation of the product-specific bioassays. And this is also why we did our first strategic acquisition this summer of a GMP bioanalytical lab, so that we have secured the supply chain for the product forward.
Thank you. Thank you, Evelina.
Fantastic answer. It's very succinct, and it sounds like you're very much on it. Excellent.
Thanks to the external experts. I think we need to a little bit try to speed up now. And Following this interaction, I think it's fair to have a two-sided interaction. Meaning, I think it would be fair if Evelina and Francesca, they would also ask or comment our external experts. So I will immediately take this opportunity to ask both our beneficiaries, as we are progressing in this discussion we're having here, is there anything in particular that you realize that, besides your successes, that you would like to explore further with either Serge, who has run a cell therapy company, or Darren, who has been very experienced in all the manufacturing? Please also think along the line on whether this kind of interaction, the one that we demonstrate today, when we had all together, is it any meaningful to you and you would like to see more of that? Please take two questions together and try to be as brief as possible. First, Francesca, thank you.
Yes, first of all, I want to take this opportunity to thank the EIC. So for the founding, but beside the founding, which of course, I mean, the founding was fundamental because they believed in our idea, which was, really at the beginning, and the innovative, but also high risk. And beside the founding, also the kind of support that we had so far, intelligent support in the sense that, for example, our annual review meeting that we have with experts outside, and that they kind of also gave us feedbacks and pushed us also to think about more challenges and also start to making us aware about the innovation aspects, the things that we have not always taken into account too much. And I think the type of events also today is even pushing this idea even further because so of course it was a great opportunity for me to meet Serge and Darren, and the discussion with Darren, for example, about opening the eyes on the thinking ahead about the how to go actually into the commercialization, into the real application, into the manufacturing process is something that needs to be essentially anticipated, and we have started discussing about it, and I think it creates essentially an opportunity to continue this discussion. And so then I think would be very welcoming. I would be very interested to continue this type of meeting, essentially kind of strategic, focused like small symposium that you have organized. And I think, of course, it will be even better, maybe less formal and even better in person, I think, for the networking part.
Thank you very much, Francesca. Very. I'm sorry rushing you. It's always the problem of time. Evelina, as briefly as possible, please, your view on the same question.
Yes, thank you so much. And I'm super happy to have gotten connected to these fantastic people that are extremely relevant for the field that we're in. And I could just, Darren, please keep educating people in biomanufacturing and bioprocessing, because there is urgent need globally for this, as you've outlined. And I think it's important for all companies in the field that there is diverse supply chain that can accommodate also novel and innovative products that can become extremely important for patients forward. So I'm happy to connect forward. And also, Serge, I would like to be, for you, I think, thank you for a bit provocative question. And I would like to turn one right back at you. And that would be going from the unmet medical needs. So you ask about different strain and different modalities. And as was discussed with Francesca, the patient doesn't care which modality, which strain. So are you, my question is, have you seen
I'm afraid
substantial improvements
very short question.
Have you seen substantial improvements in the data in the models that you've seen from this new modalities?
In seconds, please, Serge. In seconds.
My answer is that it would be more comfortable to get that discussion going on after the summit.
Okay.
Okay, happy.
Thank you very much, all. What I'd like to say is that I owe you an answer from the beginning of our session, which was among the three things that I mentioned as important for EIC, the size, that we cover many phases, and the portfolios and all that, what would be the most impactful and what would will make the difference in the future. It is my personal view that this type of meetings, which I'd like to call it as a sort of strategic intelligence in-house that we're developing at EIC, is the one that can make the difference. In other words, I don't believe that any of the three that I said, which are very obvious and very important, of course, as well, elements, constituents of EIC, the real difference can be made by building in-house strategic intelligence capabilities, Serge mentioned like that. And this kind of discussion, which is very focused, is not just a synergy, it's not just bringing people together, organizing a meeting. It requires a lot of preparatory work. It requires a lot of thinking from us. We are putting a lot of intelligence behind. You saw the projects, you saw the experts. And if we amplify that, and if we really, as we are seriously thinking at the EIC, we reinforce that this is what we believe we can make the difference. Many people can give money. Many people can give support. Many consultants. You can read reports. You can do all that. But the difference is when you really build on strategic intelligence, even where you are successful. And as the presenters showed today, even you can do that when you are at the very early stages, but also you are a company with 20 million raised funding from private and EIC. So that was the purpose, and that was what I wanted to share with you here as the key message, the key takeaway message from this session. And thank you very much for attending.
That's us done, I think. See you, Evelina. Thanks, Diego.
Thank you. Yes, will do. Thank you. Bye.
Is that you are doing?
Yes. Yes.
And what is your ideal outcome from being here today? You know, I think it's important to say exactly what you want. Yeah, what you are looking for to get, and who are the types of people
Yes. Yes.
who should approach you. I can ask you at the end how you should be approached.
My volume is. Good morning to everybody. Welcome here at the EIC Summit. Happy to join with you, hopefully the interesting next 45 minutes. Thanks to all the colleagues that organized together with us this session. And now in 2 minutes, I will try to describe what hopefully will be the next interesting session. As I was saying, we, Marco and me, are the two program managers of EIC on green technologies. And here, what we would like you to join is a 45 minutes discussion with experts coming from science as well, from VCs and from innovation fund projects. We will discuss later who are the hosts and the guests, and scientists from funded projects to show you what does it mean when we say that EIC is an hands-on agency. It means we are an agency of European Commission that has got the ambition to help in overcoming the European paradox. And how do we do this? We are something like a startup managing a lot of money, but we want to give this money to the best innovators. We want to create the culture of helping the innovation journeys of all the type of researches going from the low TRL, from the medium TRL, and from the high TRL. So going from research project to innovation projects. We look for developing these kind of things with what we call proactive or hands-on management. What does it mean? It means that the group of EIC, where we are part, the program officers, we work with the colleagues content-wise with the projects. It means we try to work with the projects, trying to help them, nurturing their needs, discussing with different type of stakeholders, both science-wise and technologies-wise. And so today we are here with the business part, with the science part, to show you how we normally try to help, try to stimulate the strategic, also strategic intelligence side of the business and innovation journeys. So coming from the idea in the lab, going to the market. And today we will do this thanks to the wonderful discussion, hopefully. And now I give the floor to Marco that will describe the topic of the today's session. Thank you, Francesco. Thank you, everybody, for being here with us. And yes, we selected this topic on hydrogen, which is a key piece of the broader puzzle to enable the decarbonization of the energy system, but also of the industrial sector and all the carbon footprint of our planet. But as Francesco said, we also would like to present our vision about the sustainable development of innovation at European level, which means to add key aspects of the hydrogen innovation journey, such as the use of non-critical raw materials and looking at the whole sustainability along the whole value chain of the production of each component of the hydrogen conversion process, but also looking at the integration of hydrogen within the broader picture, and in particular using it in sectors where the benefits can be maximized. Heavy transport, hard-to-abate industrial sector, mixing the use of hydrogen with the carbon capture
to produce renewable fuels, and addressing the specific topic of transport and storage of hydrogen, with in view in mind the key challenges to minimize the environmental impact, to guarantee the European autonomy with the supply chain of the raw products, raw materials that are needed for these processes, and looking at how where better use this hydrogen and how to integrate the storage within the whole process. For this reason, we have now, and we will introduce the first speakers. Francesco, may you want to give the floor to them?
We give the floor to we funded recently this project that started a few weeks ago. The name of the project is Let's Go. I introduce here Carles Ros that will introduce his project in 5 minutes, and then we give the floor to the next speaker. Thank you, Carles. The floor is yours.
Thank you very much. If I can see the slides, sorry. Hello, everybody. Thank you very much for inviting our project here to be shown in front of all the EIC. Our project is a Let's Go project. We come from the Catalan Institute for Photonic Sciences in Barcelona. Our goal of our project is to store hydrogen bonded to graphene oxide. Graphene oxide is a carbon derivative, as you know. And the idea of the project is to be capable to store this material in solution in water, so we are capable to have a fuel which can have the hydrogen stored in it, so we can store and recover the material. I'd like to see the slides if it's possible for everybody to follow up. Okay, thank you very much. This went too far. Yeah, this is the project. The Let's Go project is the one I'm presenting today. So the idea I was saying to hydrogenate the graphene oxide so we are capable to store the hydrogen there and then to recover it back on demand. The idea we want to face is that we need the possibility to fast fuel automobiles, fast fuel the trucks. Also, we need long-term storage, a fuel that's capable to be stored for long periods and not losing part of its hydrogen during the time. Also, we want to face this problem using materials which are not toxic, safe, they are earth abundant. And the fuel we are proposing in Let's Go and we're working now, it's a liquid fuel, it's non-toxic, and it's also safe. It's water-based, so it cannot burn, it cannot freeze by cryogenic temperatures, and it's also based on carbon and water, so it's earth abundant. As you can see in the picture in the right, we are storing this hydrogen directly bonded to the graphene structure. The idea of our project, our solution we are proposing, is to, in a single step, convert water and graphene oxide coupled with photovoltaic technology, in this case, but could be also wind technology or other renewable technologies, and in a direct step, convert it into a hydrogen-rich fuel, as a black color, as you can see here. This material can store the hydrogen for very long term, and it's in water conditions, so it's easy to store and to manipulate, to fast fuel the trucks. It's non-toxic, everything is done in liquid conditions. It's easily upscalable because we are using technologies like electrolyzers and fuel cells, which are already available for the high developed technologies. And I was saying it's a long-term storage possibilities. Our team, which is involved in the Let's Go project, it's including 6 research centers all across Europe, but also 4 industrial partners which have joined the project in the value chain that you can see in this graph. We have a partner who's capable to convert the graphite into the graphene oxide, the raw material that we use for the project. There are other partners which we are also interested in showing us, because other partners could at some point be interested in the technology, which involve into converting water by water splitting into the electrolysis and storing this hydrogen in the graphene oxide. Other partners related to recovering the hydrogen from the graphene oxide, so into releasing by fuel cell technology this hydrogen and converting back to electricity on demand, and finally on testing these fuel cell vehicles in the real world conditions. No? From stakeholders, we have identified 3 different potential markets: fast fueling of heavy transports like trucks, to be capable to use as a gasoline, easy fueling, not as batteries that really need relatively long times to recover and to recharge. For example, also maritime transportation, or seasonal or daily storage to be capable to store the energy surplus that we have from some renewable energies in electricity into this fuel to be recovered during, for example, night. Right now, we are facing the TRL increase to improve the technology, upscale it, validate it in different conditions. We are searching for financial support, both for prototyping and also for the research part, which is important in a low TRL technology like is ours, and also into explaining to society, like we're doing today, our approach, the possibilities, its low carbon cycle consequences, and also to everything related to certification of this fuel. And as you can see in the graph on the right, the main bottleneck we have is the important part of the project, the catalysis into storing the hydrogen in the graphene oxide. Because all the other phases that this project includes, it's mainly already developed in high TRL technologies. You can see it more or less here as we propose in this timeline. The idea of the project has been facing this LISGO fuel catalysis into converting the water into hydrogen stored in the graphene oxide. And because further steps could be really fast, because many of the involved technologies, like the fabricating graphene, electrolyzers, fuel cells, photovoltaic or electric vehicles, are already quite close to the market. So our project, if it's successful, with the help of all the research and the partners involved, could do a significant fast change if it's accomplished. So thank you very much for listening to us. Here you have the contact for us if you need further questions. I will be here during the Congress for the whole day, so you can find us for whatever you need. Thank you very much.
Thank you very much, Carlos. This is a very good example of a Pathfinder project with breakthrough fundamental research to explore new pathways for innovation. But at the EIC, we have the ambition to cover the whole innovation journey. For this reason, we have invited to this session also an EIC accelerator project at more advanced level in terms of technology and also market deployment of the similar approach in terms of innovative hydrogen storage, but with liquid organic carrier, so quite different technology, but to address the same challenge. So I'll give the floor now to Belen Moreno from EIC Lab that will present the project Hydrosil, winner of a EIC Accelerator grant. Probably Belen is online and should be available to present. Hi, I see you now.
Hi.
Hi,
Good
Belen.
afternoon, everyone.
So, welcome, and please, can you tell us what is the key novelty that you are proposing and why your project is so interesting? And the floor is yours. Thanks.
Sure. Thank you. So EIC Labs has actually developed a liquid carrier to transport and store hydrogen. And we are based in southern France, and we were we won the EIC Accelerator in 2020. So in the middle of COVID crisis, which was actually a gift during this rough time. So EIC Labs is a company that was founded in 2015 thanks to a breakthrough innovation of releasing hydrogen from a silicon hydride via spontaneous reaction that does not actually require any energy input. Because, well, as we all know, hydrogen will help us finish with fossil fuels and reduce the carbon print we have today, as it only rejects water when we extract the energy from it. But today, the current ways of transporting it have either a very high pressure or very low temperature. Well, any technical constraints that make these technologies expensive, or directly we don't have the infrastructure we will need, which is the case, for example, for cryogenic hydrogen. And also, of course, if we want this hydrogen to be green, we need to link its production to places where renewable energy is available and it's cheaper than other places in which we don't have this lack. In this context, the molecule hydrogen was born. Thanks to it, we can actually envisage a new massive logistics change. This new logistics change that was never thought of for hydrogen is now possible thanks to Hydrosil, which is our molecule. EIC Lab solutions will enable hydrogen transportation and storage thanks to it. But how are we going to do it? Well, via this unique liquid inorganic hydrogen carrier. As far as we know, it's the only inorganic hydrogen carrier in liquid state that is known. And this molecule is actually liquid, stable, and safe at ambient temperature and pressure, which is different for any other way of transporting or storing hydrogen today. Plus, it's also non-toxic and eco-friendly, which makes it safe for the planet. And not only can we use the existing infrastructure, such as, for example, all the infrastructure for hydrocarbons that shall stay free, but also we can reduce up to 40% of GHG emissions with it, and up to 50% of money savings compared to other hydrogen transportation solutions. Also, it has an 8.7% of hydrogen density in it, which is actually 7 times more the density of hydrogen in compressed gas, in compressed gas. Plus, also the release of the hydrogen from the molecule is spontaneous. That means that we don't need any energy input needed, and this is so unique to our liquid carrier. Any other liquid carrier would need an energy input to release this hydrogen. Our solution is definitely ideal for green hydrogen market. This is our vision. We always envisioned a molecule that would fit on the green hydrogen market. And because of its properties, we identified 4 main markets that represent 21 billion euros in 2026. And this is the selection that actually fits with the characteristics I just explained. But let's see why we say we are 50% up to 50% less expensive in a practical case. As you can see, these are 3 use cases that regroup different distances and there is technologies to transport hydrogen for these distances via different types of cargo, either by boat, either by road. And in any of them, hydroseal is the best technology that we can use for either a huge distance or not as big distance as we could imagine, for example, from the south of Portugal to the north of France. Because indeed, well, since we actually patented our molecule, we took the long way. We took a lot of time to develop this. Nothing in literature was known before. So today, thanks to the EIC Accelerator, we are about to start our Series 8 fundraising in 2022, because the EIC Accelerator founded the demonstrator of a 6 to 7 TRLs of our solution. And this fundraising will be taking place next year. We are very happy about it because, since we started in 2018, the first milestone that we are going to have is right now this large-scale demonstrator we are building, and we will further do other fundraisers in 2025 and 28 to go industrial and to dominate the industry of hydrogen transportation. Of course, we wouldn't be here today without the amazing talents we have in our team that develop around 60 patents of EIC Labs. They are all ours, and they regroup not only the hydroseal molecule, but also the process of charging and release. That's why we are kind of the first in the world of liquid inorganic hydrogen carrier. If we all wonder a decarbon future thanks to hydrogen, we need to think of EIC Lab solutions as the enabler of this game changer. Thank you very much. And I guess you will have plenty of questions, so do not hesitate to come to me and ask this. Thank you.
Thank you very much, Belén. Let's clap our hands for the very interesting presentation. And now, as we say in Italy, we serve the meal and now we start discussing and eating. So we give the floor, if possible, to Professor Chen. Are you in, Ping? Can you hear us?
Yes, I can hear.
Okay. Can you kindly, Ping, introduce yourself in 1 minute? And then the floor is yours to make questions to Let's Go or to Hydroseal or EIC Labs, so wherever you prefer. The floor is yours, Ping. Thank you.
Thank you. Hi, it's Ping Chen from Dalian Institute of Chemical Physics, Chinese Academy of Sciences. And I and Marcelo, we have been in hydrogen storage for many years, and we have also with IEA hydrogen task, we work together. So it's very nice to join this events. For me, it's very interesting to hear these two presentations. One is for graphene oxide-based material. So my first question to Thales. And I'm wondering that for this, for your system, is hydrogen release you need energy input or just a spontaneous hydrogen release from your material? As I can see from your presentation that during the reaction of your graphene oxide with water, you create carbon-hydrogen bond. So that means that your hydrogen is chemically stored in your material. Am I correct?
Sorry, can you repeat the last question, please?
Yeah.
Your last sentence.
Yes. Wondering that it's hydrogen stored in your material in a chemical way.
Is the hydrogen, Carlos, stored in the chemical way or in the physical way? Is it absorbed or absorbed?
Chemical absorption, yeah. We are bonding literally the hydrogen to the carbon.
And my also question is there, when you release hydrogen from your material, do you need temperature or do you need catalyst? How fast hydrogen can be released?
So, yeah, this is a good question. This is precisely what we are researching right now. As we were saying, this is a project we just started 1 year ago from a very incipient idea there was. Right now, we can desorb this hydrogen easily with temperature. We need results about 300, 400 Celsius degrees. But the idea is that this is a first step to understand the dynamics of the chemical desorption from our material. The idea further on is to start introducing catalysts which can lower this kinetic or this thermodynamic barrier, so we are capable to desorb with much less energy, up to the point that the idea is that this material doesn't require temperature. It can be done directly in water environment and in fuel cell conditions. So the intention is that this material can, as a liquid, touch any kind of membrane, which is fuel cell membranes, and be capable to give this hydrogen to the membrane and close the cycle, the circuit as electricity, and this way generate electricity directly from the liquid.
Thank you. And then I have another question to Berlin on silicon sealant-based material. It's very interesting that you mentioned that this is a very first inorganic solution for the hydrogen storage. So my question is that compared with the sodium borohydride solution, the water, we know that sodium borohydride will hydrolyze to hydrogen. This is a known technique. So compared with that technique, what's the advantage of your system?
Well, thank you for your question. Compared to other solutions, such as, for example, the one you mentioned, is the fact that our density is one of the highest one compared to other hydrogen carriers. It's 8.7%. I don't know for sure the percentage of hydrogen density in sodium borohydride, but I'm aware that it's a little lower. And also the fact that we can actually transport our molecule with existing infrastructures. We can actually, at the moment that we bound the hydrogen into the silicon, we create the silicon hydride that you can transport with the existing infrastructure. So we won't need to do any investment on that. And the most important part is the fact that when we are going to release this hydrogen, it's going to be a spontaneous exothermal reaction. So we won't need any energy input. That's the main difference. That's the main innovation. And the three of them make HydroSeal the game changer it is.
Yeah, and it's a very good point there. But maybe you know also that sodium borohydride also give hydrogen spontaneously. This hydrolysis is also an energy-free with spontaneous reaction. So my question is that if you release hydrogen from your system, you need to regenerate your material back to the hydrogen-rich state. So what's the energy requirement? Do you know, can you give us an estimation on the energy requirement for the regeneration of your material?
Sure. So indeed, we are going to recycle this material. We are going to transport it back to the point of what we call the charging point. And at the charging point, we are going to need more or less the same energy we used the first time to charge hydrogen into it, because the process is so industrial intensive. It's going to use anytime 40 kilowatt hours per kilogram of hydrogen.
Thank you. So Francis, my question is fun. Thanks a lot.
Thank you very much, Anne. And coming back to the overarching aim to use hydrogen where best fits, where we can maximize the environmental and sustainability aspect. I believe that hydrogen can be used in heavy transport, in aviation, in shipping, in areas where really we don't have an electrification alternative. For this purpose, it's particularly interesting to see what kind of storage technologies could also look at this potential integration and to maximize the use of hydrogen where we really need alternatives and we don't have other options. So now it's a true pleasure for me to leave the floor to Professor Baricco. Marcello, could you please introduce yourself and then tell us a bit more about your experience in hydrogen storage and in particular in metal hydrates, where you've been working since a long time, and how you see these technologies with liquid carriers, powders in the overall research and innovation arena. And of course, if you have hot key questions for our projects, they are more than welcome. Please, Marcello.
Yes, thank you very much indeed. It is a pleasure for me to join this meeting. My name is Marcello Baricco from the University of Turin. As you said, I'm playing with metal hydride since a long time. And together with Ping Cheng, we are in the International Energy Agency Task 40. As you said, I'm playing with the metal hydride since a long time. And frankly speaking, liquid is a dream for the people working in hydrogen storage. So in some sense, hearing about liquid state is a pleasure for us because this has been a discussion for a long time in the past. But because I have just 6 minutes to my time, so let me point 6 topics. The first one is, as you said at the beginning, we have to play with high TRLs. So the key point is how these technologies can be really moved to the high TRLs. And the point is, when we are talking about hydrogen, we are talking about quantity and quality. And when I'm talking about quantity, I'm talking about kilos and in some cases up to tons. So the first point is how much this technology can go into the direction of tons of hydrogen. The second point is related to what Ping was already saying, related to the thermodynamic of the system. As you say, the reactors have a spontaneous reaction in one direction, but it has to be not spontaneous on the other direction. So that is a point that has to be clarified when we are dealing with new technologies. The other point is related to the kinetics. As Pinch was saying, we need a fast delivering of hydrogen, either in uploading and in downloading. So you are talking about catalysts, you are talking about silicon. One point is to understand how much these two different steps in your technologies are related to the critical raw material. Silicon became a CRM when it is at electronic grade. So it is crucial to understand what is the purity of the silicon that you need, because this can have a high impact, in the upscaling. And we are dealing with catalysts that, to my understanding, you don't have yet in your hands. You have to take care of the presence of possible critical raw material. The fourth point is if the technology is able to take any hydrogen, because we know that right now we have hydrogen coming from the electrolysis, but we have also gray hydrogen that is running around. So how much the technology is sensitive to the quality of the hydrogen and the pressure of the income hydrogen in order to be able to say this is a spread technology for any kind of applications. The last point is related to the applications. I see in the Let's Go project that there are partners from industries. By the way, two of those partners are nearby me, so I know the system and then the companies very well. So I just like to stress the point that in such a kind of project, demonstration have to be clarified, shown. So it is crucial to understand how much this, let me say, starting point can actually be applied in the real demonstrator. And when the demonstrator are looking for hydrogen handling, they ask basically 2 points. The first is cost, and the second one is, in some sense, density. You mentioned in your websites that you are dealing with 10 times density of gaseous in one case, 7 times gaseous in the other case. So these numbers on density have to be clarified in order to attract potential investor in one sense and the applicants. That's it. Thank you very much.
Many thanks, quite a lot, but...
Yeah, we noted all the parts. As we can say, Marcello, we always see that you are a full professor, so we were listening. Carlos took all the notes. I also took the note, Carlos, so if you lose something... Belen, I hope you take all the notes. It's in my notebook anyway.
I did. I did indeed.
No. Thank you very much for the questions. They are really the key questions that the project right now is trying to face. No? We have some partners which are experts in understanding the field of the market that we have to precisely respond to, which are the questions you were giving us. No? Quantity and quality, at least we're working with raw materials, and if they are pure from their origin, so it's not a part of our project at least, but carbon, pure carbon and pure water, our product will be abundant and purely carbon and hydrogen stored in it. When we release this hydrogen, our intention is this hydrogen is pure because the carbon is a part of graphene, which is quite a strong material, so we don't expect to generate hydrocarbons together when releasing the hydrogen. You spoke also about the velocity during the process, no? Both the velocity on releasing the hydrogen from the material. We expect to do this fast, but we don't really know because we really have to work a lot on the scientific part to understand the dynamics, but here where the catalyst play a role. I will talk about the catalyst later. And, but at least we have a liquid hydrogen storage, so we intend to be fast on charging the vehicles, the long storage, whatever we do in flow cell conditions, for generating in flow cell conditions or for fueling a car, like in an actual infrastructure of fueling cars or distributing energy. No? Scarcity of materials, we are facing right now only water and carbon, so these are very abundant and cheap. But the catalyst I spoke about, yeah, it's the key role that will play a key role in the fabrication. They will develop complexity in fabricating the graphene oxide, including this catalyst atomistic incorporation in the nanoscale of the material, so to reduce both the energy balance to be releasing this hydrogen together with also the speed of this release, because it's a kinetic part of the chemical bonding. You spoke about how pure do we need this hydrogen when we store it. The good part of our project is that we are not storing hydrogen. We are generating directly from water, graphene, and the renewable energy in electricity. We are directly generating a fuel containing hydrogen. So it's as pure as the water and the carbon we inserted, but we do not require a purity on the hydrogen feed that it's given to us, because we are generating our own hydrogen from breaking the water molecule, together with a bit of hydrogen gas and oxygen, which is a side product that could be used for other purposes. And yeah, you spoke about the demonstrators, the cost, and two critical parameters, the gravimetric density and the volumetric density. These are the key parameters because right now hydrogen as a gas has a main problem. No? It's a very energy-dense material, but it's a small molecule which occupies a lot of volume, and we cannot have cars with a very large tank because then there's no space for the car. Or lithium batteries are very heavy for storing the energy, so for long travels we need a lot of batteries. There's a lot of weight in the cars which we will have to move. Yes, this is the scale of the project that precisely the project will try to face. We are expecting to beat the hydrogen gas and cryogenic hydrogen with our project, but this is a question that will be answered during the development of this project. And so for that, there are partners, experts in trying to find the key parameters for that. But also, well, here we are to expose our project, so if anybody wants to give any contribution, we are open to that.
Yeah, thanks, Carlos, for your very comprehensive reply. I will leave also the possibility to reply to Belen before coming back to some other issues related to the innovation market and so on. But please, Belen, and then we go.
Okay, so
I ask you to be quick. Sorry.
Questions. Yeah. Luckily, I also noted the questions, so I will try to answer as fast as possible. So regarding the quantity of hydrogen, of course, if we are determined to go industrial, well, we never thought of hydrogel to be used for small quantities and low qualities. Hydrogel has been a molecule that has been optimized for have the best quality, and in terms of quantity, we are actually open to the best quantities we could actually think of for transporting hydrogen via hydrogel are up to 10 kilotons. That means that we are definitely ready for massive transporting massive quantities of hydrogen. Regarding the quality of raw materials, well, and the silicon is actually it's going to become it's going to be born from silica, any type of silica. We are going to modify silica of any type of quality, and it's going to be the carrier, a perfect carrier that can actually carry any type of hydrogen. That means that even if we have hydrogen, gray hydrogen, any type, or even if we have dirty hydrogen, that means hydrogen with impurities, this hydrogen is going to be purified during the process, and at the moment of the release, we are going to only release the purest hydrogen, almost 100% pure hydrogen, which is actually perfect for example, fuel cells. About the kinetics and the spontaneous reaction, well, it takes around a few seconds to release the hydrogen. Of course, it will depend on the quantity we want to release in and the time we want to. But the only thing that will change is the quantity of thermal energy that we are going to generate from this process. But in a few seconds, we can actually release the hydrogen we have into our hydrogel. Also, regarding the applications, well, this actually is going to also answer the fact, the question of density, because, for example, one of the applications we have in mind is the underground strategic storage. The underground strategic storage is something that is done, that has been envisaged to be done via compressed gas, but it can't, because it takes a lot of place, a lot of room, and it's kind of complicated to keep it in gaseous state with no technical constraints. In our case, hydrogel is perfect for that because it's a liquid that we can isolate in underground storage and underground caves, and there won't be any dangerosity because hydrogen won't release by itself. And regarding, of course, cost, cost is the obvious question. In any case, we did the technical assessments and the economical assessments to be able to say that the ideal price that we will get once we will get industrialized will be around 3 to 4 euros of hydrogen produced and stored into hydrogel. That means that in the end, we will be able to bring hydrogen from places where it's cheap to produce, for example, where we have renewable energies, to places where it's expensive at around 3 to 4 euros delivered.
Thank you very much, Belen. And now we move from the science questions to the market and financial questions. So it's a pleasure to have Kinga Stanislavska and Alessandro Zampieri. Kinga is working in the VC world, and Alessandro is working more on the Breakthrough Energy Initiative in the part of the project. So I give the floor to Kinga to make questions and introduce herself. Thank you, Kinga.
Thank you very much, Francesco. Great to be here. Kinga Stanisławska. I am an IC member of EIC fund, and I am also working in the venture capital space for a number of years. What I wanted to ask, perhaps starting with the more advanced company, so Belen, what is the final vision for this company? What it is that it's dreaming about? Where will it go? And then the same question to you guys, although I know that you're still early in that roadmap, but always founders see that. So would love to just understand, about your vision.
Thank you for your question. So, the final vision, the one we want to accomplish in 2028, is to be hydrogen providers, because hydroxyl is a molecule that doesn't exist per se. It needs hydrogen to exist. That means that if we deliver hydroxyl, we are delivering hydrogen. In that case, when we count on making sure that 80% of the value chain of hydrogen is going to work, that means doing key partnerships with hydrogen producers, logistic companies, etc., and delivering hydrogen through these end users of hydrogen that will use either for mobility, for in the industry, for different applications, the hydrogen.
Now the floor to you, Carlos or Luis.
I'll answer. Well, just to say that I'm in the Let's Go project. I am representing the knowledge and technology transfer of it. And in regard to your question, Kinga, I think the point is that we try to represent the consortium as a whole. And we want to establish a solid IP portfolio that would enable not only the final Let's Go application to develop, but also if individually each of the members of the consortium can find an end application and exploit these patents, that would be an added value to the whole project. But eventually, if we try to focus on hydrogen and Let's Go as a hydrogen carrier, we expect to apply to further funding, even provided by the EIC or any other funding. And this is really why we are in this place, which we are open to all the discussions, to all the criticism, and try to see where the technology can develop, and what is the trend in the market, and what is the trend in funding. This is, we need to know just to see where the next 3 years will take us.
Okay, both of your companies are looking in terms of EIC Labs. It was already in the presentation, but both of your companies are looking ultimately for financing from either corporates or from the venture capital market, angel investors and other institutions. And in terms of that, in addition to money is important. What is it that you're expecting to get from your investors? What kind of smartness do you think could add to what you have to build the company together and to have success? Maybe starting with you guys here.
No, yeah, that's really also another very important factor, because as scientists, we are used to maybe focusing only on the science, and bringing this into the market requires a completely different mind and a complete different set of skills. So I think when you have investors working along your side, interested in your project, understanding your technology, you really learn much more from them than from the scientists, because you're not concerned anymore with the science. We're concerned about selling the technology and really getting into the market. So the ideal contribution or collaboration with VCs or other type of investors would be to them working along you, understanding your technology, and this way learn how to develop it and sell it. This is basic for us researchers that sometimes don't understand or don't know how to communicate correctly to the investor world.
And if I can add a little thing as a scientific in this part, it's very usual to see that the scientific wants to get deeper and deeper and deeper into the knowledge, and somebody has at some point to put a stop or to redirect the direction. What is less costly? So let's try to first search for catalysts which are less costly. Let's aim for materials which are less costly. This approach, sometimes the scientist doesn't spend too much time in researching where really the cost is an amount of knowledge which somebody has to learn or not. We have learned as scientists how to do the thing, but maybe not in which context completely to do that. So it's very important always to not just do science by science, to do science to transform the society, to transform the world, because we are facing some problems as a society that we really need to face scientifically.
Okay, thank you. And over to EIC Labs.
Okay, so from our side, our seed fundraising was already done with investment funds, and we are very thankful to them. But for this new Series A, as it's going to allow us to go through to industrial, we'd love to count on all, apart from the financial support, with the industrial expertise. That means either expertise in industrial processes, either expertise in hydrogen, but in any case, we are not only looking for financing, we are also looking for partners. We are looking for those who are players in the value chain that need a solution like ours and that would be interested in developing it at industrial scale.
Thank you, Belen. I would like to leave the floor also to our last expert, Alessandro Zampieri from Breakthrough Energy Catalyst, that could provide a vision about support schemes and how the venture funds see this kind of entrepreneurship investments, and what is your vision and how you would like you could support such innovations from your point of view. So please, Alessandro.
Hi, everyone. Thank you, Francesco and Marco, for having me. Very interesting panel. Congratulations, Belen and Carlos and your teams at EIC Lab and Let's Go project. Very interesting technologies, essential to enable a hydrogen economy and being able to connect demand and supply by storing and transporting hydrogen in a cost-effective, efficient and safe way. So very interesting panel. I would be, what we do at Breakthrough Energy Catalyst in a few seconds. Basically, we are an investment fund, clean tech. One of our technology of focus is hydrogen, in addition to long duration energy storage, sustainable aviation fuels, and direct air capture. And we look at supporting projects that are early commercial. So projects that are not necessarily in the money because of the green premiums of these technologies today. And we help them with what we call a blended finance approach. So we come in with offtake agreements, with sub-market equity and philanthropic grants to support this project and de-risk them, reach financial closure, close the financing gap, and transition into implementation. Our aim is to actually help scale up and roll out technologies, deploy technologies at a much faster rate than what the solar and wind industry have done maybe 2-3 decades ago, simply because we don't have the time and the luxury of time, to wait so long to see such technologies deployed in the market, which are essential for decarbonization of industries. So that's what we do. Maybe if I may, Belen, first a question to you. You spoke about a demonstration project. I would like to hear more about, if possible, about the size that you have in mind, where you stand in the process, and what's the largest proof of concept that you deployed so far, if you can speak about that.
Sure, no problem. So regarding this demonstrator we are going to have in mid-2022, it's actually a demonstrator at the minimum scalable size to industrialize the process. That means that it will have a non-commercial quantity of production per day, which is around 1 kilogram. Of course, we are not aiming to sell this because that wouldn't make sense, but it would allow us to do all the tests that we need to prove that the molecule is safe, etc. Because, well, of course, as when you have a new molecule in the market, you need to do several tests, etc. And also, it will allow us to integrate the first projects. Of course, we are into collaboration projects today already, but most of them are theoretical. with this demonstrator, we will be able to go practical and to start finally manipulating with our partners the molecule, because the proof of concept we have today is just lab scale in our labs in our headquarters in Aix-en-Provence in France. So the idea would be to at least start improving this also with our European partners, of course.
Thank you, Belen. And what do you expect to be the challenges in the development of this pilot project? And then I would also like to hear more about the timeline to see the next scale project, if you want, so kind of pre-commercial, early commercial scale.
So the main problem that we already face it and we will be facing is the fact that when people don't see the machine, they don't think it's going to work. So as long as we don't have the industrial facility, it will always be very hard to make people aware of the fact that it will work. Even if we have it at large scale, as long as we don't have it at industrial scale, they won't believe in it. And our next step after this would be a more consequent pilot of around 10 tons of the solutions, which is around a ton of hydrogen per year. That will be an industrial, what we could call an industrial pilot. It will be in 2025. And this pilot, of course, will be integrated into the Series S fundraising with the idea of precisely avoiding this problem, because the advantage, let's call it like that, of starting the research in 2018 is that with this demonstrator, all the risks will have already been taken.
Got it. And sorry, I didn't get the scale of the pre-commercial project, the last one that you mentioned, 2025.
So from 1 kilogram to 1 ton.
Sure.
From the kilogram to the ton.
From kilogram to ton, Alessandro.
Yeah.
Okay.
Thank you very much, guys. Sorry to interrupt, Alessandro, but the time, as usual, is terrible. So it's as guys from the financial teaches us, the time is stringent, and so we have to go to the next step. And so I would like to thank everybody. I hope that you have seen an example of the program managers try to work, driving the research in the direction of the innovation, speaking about corporates, about strategic, as our colleague Dano said before, about strategic intelligence. And just to give the floor to Marco to conclude, just to say that we are here speaking about hydrogen because we in EIC have got projects funded in hydrogen, so what we call thematic portfolio on hydrogen, and we are starting to evaluate in the next days a call that Marco and me did, a challenge-based call on Pathfinder on green hydrogen production routes. So thank you to everybody. I give the floor to Marco to say the last words. Thank you.
Yes, just to say that we are really on this topic of storage, energy storage, so we are working on other specific challenges on mid-long-term storage, which is very complementary to hydrogen, and also the use of hydrogen combined with carbon to produce renewable fuels, which is another way to maximize the use of this technology. So I thank you for your attendance. Thank you, everybody, and for sure we will go on with this discussion further soon. Thanks.